Okay, we are good to go. So, my name is Nir Goldstein. I'm the Managing Director of the Good Food Institute here in Israel. Welcome to our event about cellular agriculture and harnessing fermentation to feed the world. We are very excited to have you here with us, even though we have this very challenging event that affects all of us. So thank you for clearing your schedule for something like 2 hours of escapism in which we'll be able to talk a little bit about proteins. Just a few technical issues before we begin. We have a Q&A button here at the bottom. You can use it to present questions to our speakers. We will have a Q&A session at the end of the two presentations that we have for you today. Today's webinar is also recorded for a future use so that you could be able to see it later on. We will also send you the presentation in an email following today's event. Today's agenda, we'll have a brief introduction from our partners for this event. Then we'll have David's presentation followed by Ilan's presentation, Q&A as I mentioned, and a few resources. So we have a full schedule for today. And now it is my honor to introduce you to Dr. Gil Jonah. Hi, Gil.
Hi, everybody. Thank you for joining us. And let me introduce myself. My name is Gil Jonah, and I'm the head of the Bacteriology and Genomic Repository Unit here at the Weizmann Institute. I'm here since 2005. Again, thank you for joining and today with this Zoom online seminar. I was really hoping to host you here around in our marvelous campus, but the time and the era is definitely not suitable for this. So another short background about myself. Here at the Weizmann, we supply a lot of fermentation services in the range of 1 to 100 liters using 6 bioreactors, both for in-house as well as for external projects. And we also perform different upstream process development, mainly for internal projects and for protein expression. The second activity is related to small-scale bioreactors. We have 8 of them that we are using for more specialized research-related projects, such as in vivo evolution, competition and perturbation experiments, things that you might have seen recently on the use of bacteria that can consume carbon dioxide as their carbon source on cell. So thank you again for joining us, and thanks for the organizer, for Nir, David, and all the other team. And Nir, I'm passing the podium back to you. Thank you.
תודה רבה, Gil. And now it is the turn of Dr. Ronnie Bronstein, chairman of the Israeli Fermentation Society. Hi, Ronnie.
Hi, hello everybody. Let me first introduce myself. I'm Ronnie Bronstein, the CEO of BioDalia Microbiological Technologies. We are located in Kibbutz Dalia in the north of Israel. BioDalia is a manufacturer of crop protection biopesticides, and in addition, we give our fermentation services to many other companies in different fields. We have 2 big 25 tons big fermenters and the complete upstream equipment. First of all, let me thank you all for your participation in this special webinar. I wish to thank Or Benjamin for her initiative and the energy to make this seminar happen. Many thanks also to the Weizmann Institute and to the Good Food Institute, and of course to our lecturers, Dr. David Welch and Dr. Ilan Samich. The Israeli Fermentation Society is a non-profit organization which was founded in October 19 by Dr. Moti Ravhun and by myself. We have today around 80 members. Among our goals are promoting the Israeli fermentation industry, assisting academy and R&D institutes in developing new projects based on microorganisms, as well as holding conferences and meetings, as we do today, in order to establish and strengthen professionals' interactions and exchanging information. I ask, of course, everybody of you to become a member of the Israeli Fermentation Society, and more than that, to become active in our organization. I wish to thank you all the panel members, to Nir and Hadass, and wish we all successful webinar. Thank you very much. And I pass it on to Nir, please.
תודה רבה, רוני. And now it is my honor to introduce you to GFI. So who is GFI, the Good Food Institute? We are working worldwide in order to change the way we produce food from animals. We believe the animal agriculture has several deficiencies, such as it harms for the environment, for human health, as we see nowadays, and also, of course, for animal welfare. Now, many people have been trying to solve these problems over the years, but we found out that people are sticking to the way that they eat food, to the food that they like to eat. So what we are doing is we're trying to make the similar foods, but in new ways. We are harnessing the power of food innovation and also the market and technologies. We try to accelerate technologies such as plant-based, cultivated meat and fermentation in order to make meat, eggs, dairy, and seafood in alternative ways. We do it in three main work streams. The first is science and technology. We are working with scientists and researchers all over the world, and we assist them with hands-on knowledge and experience. We also have an open source fund. We are currently funding research around the world. We just announced this week another investment of about $4 million in research, including here in Israel. We are funding research in about $1 million nowadays across the Israeli Academy in institutions like the Technion, Tel Chai, Hebrew University, and more. And we also have a lot of resources for students. We have a student body in the Tel Aviv University, and we have the first course on alternative protein in the Hebrew University that is given by our own Dr. Tom Ben-Ariel. Regarding corporate engagement, we are working with the biggest food and agriculture companies around the world, also pharma and equipment companies, in order to support them in engaging with this new field of alternative protein. We are also working with investors. As most of you probably know, this is one of the hottest fields in the investment landscape. And with entrepreneurs, we have resources for entrepreneurs. We assist them in finding resources and human capital and more. When it comes to policy, we are working with governments all around the world, also here in Israel. On the one hand, we are working with governments on accelerating public funding of research in our field. And on the other hand, as all of you probably know, working on regulation of new food, which is sometimes very challenging. We are a non-profit, as you can see here at the bottom. We have a seal of transparency from GuideStar, which means that we are a rather impactful non-profit. All of our income comes from philanthropy from all over the world. And we are located right now in the US. This is our home base, but also in Brazil, India, Europe, currently in London and Belgium, Asia Pacific, which is currently in mainland China, Hong Kong, and we have some staff in Singapore and Thailand. And here in Israel, we are working to bring the Israeli innovation to support this global revolution of the food system. And we are very excited to be having this series of events, which concludes today with this webinar, in order to further engage with the Israeli food ecosystem. And now it is my honor to introduce you to Dr. David Welsh. David holds a PhD in plant developmental cell biology. He has vast experience from the life science industry, where he held various positions in business development, marketing, and product management. David joined, luckily for us, he joined GFI in 2017. And now he is the director of science and technology, where he oversees a really impressive team of scientists. So David, thank you for waking up so early and being here with us.
Thank you, Nir, and thank you everyone for joining the webinar today. Let me share my screen. All right, I'm sorry I couldn't be with you in person in Israel, but I'm really happy that we were able to do this virtually, and to allow me 40 minutes or so to talk to you about why I think and we at GFI think fermentation is the future of the alternative protein industry. Nir mentioned that a lot of our work centers around identifying white space opportunities, and there are many of these specifically related to fermentation that I'll speak about today. And we identify these white space opportunities and then address them through a number of different activities. Nir mentioned our competitive grant program. We fund more and more research in this area and also find external funding opportunities for research. So if there are any of the ideas I talk about today spark an interest in you for a new research project, I'd encourage you to reach out to us for help with funding. We also advise companies, governments, and universities on where to allocate their research and expertise. We produce technical papers and deep dive analyses on these. So if there are commercial ideas that come to mind as you sit in the webinar today, please reach out to us for support with this. And then finally, we're very interested in creating new research centers in Israel and elsewhere around the world that can focus their activities and expertise on alternative protein research, especially using fermentation technology. And one of the big questions that drives our work and the thinking around these white space opportunities is the question of how we feed the 10 billion people that will live on a planet by the year 2050 in a way that's sustainable, efficient, and safe. And if we look at our current food system, particularly the use of industrial animal agriculture, there are a number of risks that will likely prevent it from being the primary use of food to feed those 10 billion people in the future. I think you're all familiar with the greenhouse gas emission impact of industrial animal farming. It's the #1 user of water on the planet. It's a primary cause of deforestation, particularly in areas like the Amazon rainforest. You may not be aware that it's also the #1 user of medically important antibiotics. And recent research suggests if that goes unchecked and we don't solve these issues, it could cost the global health industry up to $100 trillion by the year 2050. There's health impacts as well, particularly in the area of foodborne and zoonotic diseases. And this can have an enormous financial impact that I think, as we're all well aware. And a good example of this is swine flu that's been impacting the world, and particularly China, over the past couple of years. Numerous outbreaks, over 100 in China since 2018. Over half of their breeding pig population has either died or was culled. And on the graph on the left there, I think you can see that as the pig population decreases, the price of pigs and pork goes up considerably. So this industry, or the industrial animal agriculture industry, is really ripe for disruption because of the impact that these types of events can have on the food system. Despite all of this, demand for meat continues to grow and shows no signs of slowing down. As our population increases to that year 2050 and grows by 30%, we won't see a comparable increase in the demand for meat, but much higher. In the case of poultry, that demand is expected to go up by 120%. So really significant increases in the amount of meat the world is wanting. And a lot of this is driven by income as well. As consumers move into higher income brackets and their disposable income increases, they're likely to demand and consume more and more meat. So we can tell people to eat healthy dishes like this, full of greens, grains, and beans. But what we see is that most people don't want to do this, at least for every meal. People like the taste of meat, eggs, and dairy products. And so our theory of change at GFI and the alternative protein industry as a whole is to borrow this phrase from Y Combinator and make something that people want. And really just to help the industry make meat and eggs and dairy products, but in a better way. And a lot of this innovation is not being driven by the vegans and vegetarians of the world, but really by the omnivores and flexitarians that are becoming more and more aware of the impacts of the animal agriculture industry on our planet, but still like to eat meat, egg, and dairy products, but want to do so in a more environmentally and socially conscious way. And that's really driving the creation of these biomimics, where the taste and all the other sensory attributes of these products are replicated, but by using different and more sustainable sources. And this industry, this alternative protein industry, is expected to grow very quickly. One of the most optimistic outlooks from A.T. Kearney, a market analytics firm, suggests that this industry, which would comprise both the plant-based meat industry and cultivated meat or so-called cultured meat, is going to grow to occupy 60% market share of the global meat market by 2040. So very optimistic outlooks, but if all variables point in the right direction, as this axis of uncertainty by another analyst organization, Jefferies, shows, it seems likely that we could get there. So we can continue along the path that we're currently on and creating better alternative protein products, better plant-based meat and cultivated meat products using fermentation to innovate. And we're still likely to see, even if some of these other factors don't come into play, a market that's worth over $100 billion a year. But if certain other events happen, like the meat tax that Jefferies is forecasting, then this industry can grow much, much bigger. The meat tax will cause the price of conventional animal products to go up, and at the same time as the technology improves for alternative protein products and the price of those come down, consumers grow more accepting of these new products. Jefferies forecast that we could see an industry well over $400 billion by the year 2040. So lots of growth opportunities for this alternative protein industry, and I think also a major role for fermentation to play in this new and exciting industry. When we look at alternative proteins at GFI, we categorize them into these three technology areas from a production cost and infrastructure perspective. We have proteins that can be made from plants. We can use fermentation technology, as I'll speak about today. And we can also use animal cell culture, as I mentioned earlier, to produce actual meat from animal cells. And what I'd like to hopefully convince you of today is that fermentation is central to the future of all of these areas by either producing standalone protein sources or as a technology that can provide ingredients and enablers to the plant-based and cultivated meat industries. And that ultimately in the future, we won't see these three technologies as separate industry segments, but rather as one large industry where we produce animal product alternatives along a spectrum. And we have some fully plant-based products. We have some fully cultivated or synthetic products like synthetic gelatin or egg proteins. And then in the middle, we have this large and growing segment where we're creating products that are hybrids that use plant ingredients from fermentation, microbial fermentation technology, and even cultivated ingredients, so that we might have in the future a plant-based burger that has cell-based fat or cultivated fat, but also uses flavoring or other functional ingredients from fermentation technology as well. So now I'd like to step through how we can use fermentation. And this slide is a very high-level overview. I think most of you on the call will be familiar with how fermentation is used, but just to sort of ground us, this is typically the process that's used. So we're selecting some type of source material. It could be a fungus, a bacteria, an algae, or even a plant in some cases. We're optimizing that in some way, potentially through genetic engineering, so that we have a strain that works well for a particular application. We're then fermenting that in some type of tank or bioreactor, along with a feedstock that is optimal for that particular strain. And then the end product, at least in the alternative protein category, can be segmented into these two broad categories. One is biomass, where we're using the product of the entire microbe or cell to create a large amount of protein that can be used in a final product. Or we might be using synthetic biology or recombinant protein technology to produce an individual functional protein that can be used as an ingredient. And we're starting to see a lot of new entrants into this fermentation, into the fermentation part of the alternative protein category. In the biomass area, you can look at these products as either producing protein biomass as an ingredient, and you see some examples of companies in this area down below, like Solar Foods and Air Protein, that are using bacteria to consume CO2 and other side streams as fuel to produce protein ingredients. And then moving upwards, there are now really interesting companies, corn obviously being the pioneer in this space, and many of you will have seen and even tried their products, but new companies emerging as well that are using fermentation, specifically of filamentous fungi, to produce whole cut products that really biomimic meat. And then on the right, there are a number of companies forming or that have already formed that are using fermentation to produce specific functional proteins, from dairy proteins that I'll speak about a bit later in my talk, to egg proteins, gelatin, and even companies like Motif Foodworks that are creating a platform that can be used to produce hundreds, if not thousands, of different functional proteins. So lots of opportunities for fermentation. And what I thought would be interesting today is to kind of talk through a specific food example and ask the question, can we reverse engineer a bacon cheeseburger, perhaps even a kosher bacon cheeseburger? I'll let you be the judge of that at the end, using fermentation technology. So if we start with the main component of the burger, the beef, one of the core questions that faces the plant-based meat industry is, how do we persuade these globular plant proteins that will be the primary ingredient in a plant-based burger? How do we persuade them to act like animal proteins, to be fibular in nature, to be cross-linked, so that they provide that texture, that mouthfeel that consumers associate with beef when they bite into it? So currently today, the industry primarily relies on mechanical methods to do this. In most cases, the industry is taking plant protein concentrates and isolates and putting them through a twin screw extruder, which imparts high pressure and temperature to those proteins to restructure them to create that textured vegetable protein. We're seeing innovations in other mechanical methods, such as shear cell technology and even bioprinting. But I think one area that has been somewhat explored, but where there are a lot of white space opportunities, is the use of chemicals or enzymes to modify proteins. I'm showing one example here where, which looks at not plant-based meat applications, but using soy protein to create a pudding, and the impact of cross-linking on that soy protein concentrate and the texture of this pudding. And so what you can see on the right of this graph is that when the protein was cross-linked, it created a harder texture, a harder soy-based pudding. And this created differences in the texture as well, as you can see from the micrographs at the bottom. So there's some work that's been done to use enzymes for this purpose, both with conventional animal proteins such as whey and others, and also with plant proteins like soy in this case. But I think this area is really ripe for innovation, to think about the types of textures we're trying to create with these modern plant-based meat, egg, and dairy products, and how we can use an enzyme or combinations of enzymes to create different types of cross-linking, different types of textures that could either be used as a standalone method to create these textured vegetable proteins, or used in combination with a mechanical method such as extrusion or 3D printing. And obviously, there are a number of companies that already exist that create enzymes using fermentation. Some of the big global players I've listed on this slide here, and there are others, many others, and some specifically in Israel as well. So it might be that these companies like Novozymes create novel enzymes specifically for plant-based meat, egg, and dairy applications. But I think there's also big opportunities here for existing companies in Israel and even startups to think about the creation of novel enzymes specifically for this industry. So we can use fermentation to produce enzymes to modify textures. We can also address some of the other key sensory challenges facing the plant-based meat industry. We can optimize flavor and functionality through unique additives. We can even recapitulate the experience of cooking and identify the aromas that are produced during the cooking of a burger, say, and produce those through fermentation. And even novel compounds to improve the taste and texture through fat encapsulation could be created through fermentation. For this lecture and our question of creating a bacon cheeseburger through fermentation, I'm going to talk about the first category here of novel flavor compounds that can be created through fermentation. And the example I'll use is the Impossible Burger. So you can see here a list of the ingredients in the Impossible Burger. It has some typical plant proteins, some functional additives like methyl cellulose, and other starches to bind, some coconut fat and sunflower oil, and some nutrients added as well. But one of the key additives is this soy-like hemoglobin that I've circled in a box there, that imparts that red color and meaty flavor to the Impossible Burger. So what is soy-like hemoglobin? Well, like hemoglobin is a globin, which means that it's a globe-shaped protein, and it's found both in animals and in plants. The one you're probably most familiar with is hemoglobin, which is found in your blood and transports oxygen from the lungs to the rest of your tissues. It also gives blood its red color. And you can see in this picture here, in a cross-section of a root nodule, that soy-like hemoglobin is also red. And it's red because of the way the chemical bonds bind iron and the oxygen reflect light. So this is a plant globin, and the team at Impossible Foods was able to identify this, as I said, in root nodules of soy and other plants, but it's difficult to extract. So what they decided to do was to synthesize this using recombinant protein technology as well. And they now produce this via yeast as one of the key ingredients in the Impossible Burger. So I think, again, here there are lots of opportunities to look at other compounds that might be important in the taste and flavor of meat, and identify these compounds through other non-animal sources, and then synthesize them through fermentation technology to provide new and better ingredients for the plant-based meat industry. So we might, I think, be able to solve the question of biomimicking beef through plant proteins and novel ingredients from fermentation, but perhaps we might need cultivated meat technology as well. Maybe there's a missing taste or texture that we need to use animal cells, either to create muscle or fat or some combination of those two that can be then added to other plant-based and fermentation ingredients. But what's the role in terms of cultivated meat and fermentation? How will fermentation play a role here? Well, one of the main challenges facing the cultivated meat industry today is the requirement in the future, when this industry becomes commercial, of very large volumes of inexpensive raw materials. Today, in order to grow these animal cells to produce meat in a way that doesn't require animal components, so using these synthetic cell culture media, the cost of these is quite prohibitive. And currently, surveys show that the cell culture medium makes up about 60 to 90% of marginal costs of all of the cultivated meat companies. So how do we bring the cost of these really expensive synthetic growth medium down to less than $1 a liter, so that to make cultivated meat price competitive? Well, if we look at the ingredients in a typical serum-free medium formulation, it's going to be a combination of inorganic salts, amino acids, vitamins, and other nutrients. All of these that you see in the green boxes, we can convert these from pharmaceutical grade to food grade components and lower the cost. Many of these, I should add, are currently produced through fermentation technology as well. But it's really this group of growth factors or proteins in this orange box in the middle where fermentation can play a big role. Work done by Dr. Liz Specht at GFI to look at how we can reduce the cost of cell culture media and whether we can bring it down to less than $1 a liter shows indeed that we can, by reducing the concentration of these growth factors that are present in synthetic cell culture media, either by training the cells to need less or by improving the efficiency of these proteins so that we need less of them in the growth media, and by scaling the production of some of these growth factors so that the costs come down. And then moving, as I said, all of the components from pharma grade to food grade, you can see by this paper exercise, we can bring the cost down of the cell culture media to significantly below $1 a liter. And what's interesting is that now is that a lot of this work has now been validated by the Paul Burridge lab in the US, where they did just that. They did some growth factor engineering. They optimized all the concentration of the ingredients inside cell culture media, and were able to reduce the cost of cell culture media down to less than $10 a liter, so a 97% cost reduction. So I think this is nice because it shows you that theoretically we can bring the cost down, but practically as well, even by just doing this work in a lab. So I think here again, there are a lot of opportunities in the fermentation industry to think about how we can re-engineer these growth factors and then use microbial fermentation to produce large amounts of these for the cultivated meat industry. And when I talk about large amounts, what do I mean? Well, if you think about the cultivated meat process and going from a small vial of cells where you need to freeze and aliquot those cells out for production runs, to the seed train process, to the final large scale culture of animal cells to produce cultivated meat, we're going to need a large amount of manufacturing facilities and cell culture media to fuel the production of these cells. We estimate that one 20,000 liter bioreactor can produce roughly about 175 metric tons of meat per year. And if we look back to that A.T. Kearney market projection, whereby 2030 cultivated meat will occupy 10% of the global meat market, that's about 35 million metric tons of meat per year, which would, if we had facilities that had 20,000 liter bioreactors, we'd need about 10,000 of those globally. So a really large number of facilities. But where this relates back to fermentation and those growth factors I spoke about is the amount of media that this industry is going to need. So in the region of 50 million metric tons of powdered media, all of which containing fermented ingredients such as amino acids and those growth factors I spoke about. So again, really big opportunities here just in the cultivated meat space. So I think we've solved the question of beef. We can make it through plants and fermentation, potentially even using cultivated meat technology. What about bacon? Can we make bacon through fermentation? Well, I think the answer is yes. So I touched on this before, but there's a number of companies now that are using densely structured filamentous fungi and fermenting that to produce meat-like texture. We've seen corn innovate in this area for a number of years, but now there's some really exciting startups in the US and elsewhere that are taking this technology to another level. The one that I'll highlight here is Atlas Food Co. And I think in the picture there, even though it's quite small, you can see that their bacon prototype looks pretty much like bacon. And I've heard, although I haven't had the opportunity to taste it yet myself, I've heard that it tastes a lot like bacon as well. So really exciting innovation here, just using fungi in a novel way to create that texturized meat, particularly in the area of bacon, to create that that part of our bacon cheeseburger. All right, so we've got the beef, we've got the bacon. What about the cheese? How do we innovate, and how do we use fermentation to create better cheese analogs than exist on the market today? Well, many of you might be familiar with the overall process of making cheese. In fact, you probably know that one of the key parts of cheese making relies on an enzyme called rennet, which is for the most part produced through fermentation technology. That enzyme rennet cuts off a specific fragment of one of the caseins, namely kappa casein, which then causes these casein micelles to move into the secondary stage of cheese formation, where this cleavage permits the aggregation of micelles, and a gel-like structure forms, which is the beginning of cheese. So how can we use fermentation to create these structures? Because currently, I think if you've sampled any of the plant-based cheeses on the market, I think you would agree that they don't have all of the functional characteristics of a conventional cheese. So we can think back to using enzymes to modify plant proteins to create these types of micelle aggregates using plant proteins, or what I think a more promising area is to apply fermentation and recombinant protein technology to create actual dairy proteins as then inputs into the cheese making process. So there are a number of really interesting startups in this area as well. Perfect Day and New Culture in the US, and Legendary in Germany, and a couple of stealth companies as well, using this type of technology to recombinantly produce dairy proteins. I had the chance to sample Perfect Day's ice cream that uses recombinant dairy proteins as a primary ingredient last year, and was really amazed by how similar it was to conventional ice cream. And so I think in the next couple of years, we'll see some really interesting improvements and innovations in the use of these recombinant dairy proteins to create different types of cheeses. But just 3 companies here. So again, I think really big, wide open space to think about how we can use either enzymes produced through fermentation to modify plant proteins to create better plant-based cheeses, or to create actual cheese using recombinant dairy proteins as well. So what I've shown you by this, I think, is that yes, I think in the future we will be able to reverse engineer a bacon cheeseburger using fermentation as that central technology, because fermentation expands our ability to innovate with both plant proteins and mammalian cells. Enzymes, as I mentioned before, can improve protein functionalization or coagulation by restructuring those proteins. We can use fermentation-derived ingredients to enhance the flavor or the texture of a plant-based product. We can use fermentation to produce growth factors that will aid in the production of cultivated meat. And then we can even use fermentation to produce 100% derived, fermentation-derived ingredients, such as the synthetic dairy proteins I mentioned on the previous slide. And I think the really exciting part about fermentation is how quickly it can disrupt specific food ingredients. This graph here from a report done by RethinkX shows you a couple of examples. Rennet, I mentioned, which is the enzyme used in cheese production, riboflavin, and non-animal insulin, which is used as a therapeutic. And you can see as the recombinant versions of these proteins were introduced into the market, how rapidly they took over as the primary source. And what we're likely to see is that the market capture of these recombinant ingredients, protein ingredients created through fermentation, is going to depend on the structural complexity of the protein in question. So if it's we're likely to see more simple type proteins introduced in the coming years and less complex ones later. Concentration plays a critical role here. So if the protein is only needs to be present in the final product at a low concentration, we'll see more of those entering the market in the coming years. And then scarcity as well. If the conventional ingredient is not found easily, we're likely to see those recombinant versions of those ingredients enter the market sooner rather than later. And this plays into the question that we get asked often is how price competitive can alternative protein ingredients be in this using fermentation technology? Will it face some of the same challenges that the biofuel industry has faced? And I think this really goes back to the concentration question on the previous slide. So if we're talking about using fermentation to produce ingredients that may need to be at a 30 or 40% concentration in the final product, it might be challenging to get the cost down to a level where the end product can be price competitive with the conventional product. But if we're talking about lower concentrations, particularly in the next 5 years, I think the technology and the advancements we're already seeing in this space indicate that it's very possible to get the cost down to an area where it's going to be price competitive. So right at the beginning of my presentation, I talked about some of the white space ideas that we've been working to identify across the entire alternative protein sector. But what I wanted to share with you now is some of the most promising and I think most impactful ideas that we've identified specifically related to fermentation. And I've outlined these on this slide here. So I'll start on the left with some commercial ideas related to host strain engineering and even end products or different product lines. And this is using fermentation not just to create new protein ingredients or new flavor additives, but as a way to create new lipids and binders for the plant-based meat industry. If you remember back to that slide where I talked about the Impossible Burger, that uses coconut fat as one of its primary fat sources. Probably not the most sustainable fat source. And if we think about how rapidly this industry is going to grow, we're going to need a wider toolbox of fats for use in these plant-based meat, egg, and dairy products from a sustainability perspective, but just a resource perspective as well. That hopefully will also have different functional attributes too. And I think this is where fermentation can play a really critical role of identifying the types of fats that we need to provide the appropriate nutritional profile for a plant-based meat product and the right functional characteristics, and then engineering these and then manufacturing them through fermentation. As this industry grows and we identify novel microorganisms that can consume waste streams from other industries, or even the food industry as a fuel source, we see a big opportunity for a marketplace for fermentation feedstocks where innovators in this industry will have easier access to waste streams and byproducts and other feedstocks to fuel the microorganisms that they're growing to produce alternative proteins, fats, and other ingredients. If you think back to my example about heme and what Impossible Foods is doing with that, and then think about all of the compounds that are present in different types of meat products, there's huge opportunities here to identify and better understand these biosynthetic pathways that produce these compounds in meat, and then producing those through fermentation, just like Impossible Foods produces their soy-like hemoglobin through fermentation. And then finally, we can also use fermentation to enhance plant-based meat, egg, and dairy products. So think about how we already use fermentation to create products like kimchi and sauerkraut and tempeh. If you think about how quickly the plant-based meat industry is growing in terms of the number of crops and the number of proteins from those crops that we have access to, and then thinking about how quickly we can grow the number of microorganisms that we're working with, and then combining those two sources together in novel ways to create better and better plant-based products that have improved texture and improved taste. I think you can again see that there's really wide open opportunities here for more innovation, more companies, and more research. So I talked a lot today about the market for alternative proteins and some of the science behind it. If you'd like to dive into any of this in more detail, we have a lot of resources on our website, services for the industry, from more detailed market analyses, deep dives into the science, and even policy briefings, and we can help you make connections as well. And I'd recommend that you head over to our resources page at gfi.org/resources to learn more about all of these resources. Or you can reach out to me. I've put my email address here. I'd love to stay in contact with all of you and answer any questions you have or help you with any ideas you might have generated from the presentation today. And with that, I'll hand it back to Nir. Thanks, everyone.
Thank you so much, David. Please don't go anywhere. We will be taking questions after Ilan's presentation. And with that, I would like to present Dr. Ilan Samish, CEO and founder of Amai Proteins. Hi, Ilan.
Hi. And thank you, GFI, Weizmann Institute, and the Fermentation Society for hosting me in this webinar, which I wanted to give at Weizmann, which is was my second home for many years, but here we are at the time of Corona. And Corona is all about proteins, and we are talking about proteins. My talk will not be a comprehensive, well-balanced talk like the one of Dr. David Welch, but rather a collection of thoughts and challenges about bringing a precision fermentation protein to the market. And I'd like to open with a disclaimer. I'm the founder and CEO of Amai Proteins, which makes designer proteins by precision fermentation and has fee-bearing agreements with several large food and beverage companies. And this talk expresses only my own opinions and not that of Amai. And it's not also graphically polished as the ones that I give on behalf of Amai. So just in a word, what Amai does is to fit proteins to the mass food market, and we have computationally analyzed a bunch of proteins, meat, milk, and plant proteins to make them tasty, stable, cost-effective, digestible, hypoallergenic, sustainable, and of course environmentally friendly. Our main protein, which is now close to the market, is a hyper-sweet protein which replaces sugar. And at the end of the day, what we make is food. We make food with a lot of collaborations. And when people ask me why I enjoy my work so much, I show them that part of my job is to be part of the super taster panel of tasting sweetened beer, and then they all understand why I love it so much. We have a bunch of big company partners which also help us quite a lot, but I would like to move to the substance of this talk, which is what are some of the fermentation challenges to bring a protein to the market. And for that, you need a protein which is fit for the mass food market, including when it is produced by fermentation. It has to be functional, it has to be structurally stable. You have to have for that also a high yield expression of that protein to make it profitable. The media is only one component. Another component is how many grams per liter do you make. And then in order to get it to the market, unlike what is a current situation with meat that is loaded with antibiotics and many contaminations, you want to have it with no antibiotics and no contaminations. So the standard that you're asking for proteins coming from fermentation is far, far above that you're asking from what is coming from the farm. And on top of that, you want to be cheaper than what is coming from the farm. And David spoke about the issue of media cost, growth, time, and energy using aerobic or anaerobic fermentation is another issue, and the purification system that you're using. You have to have a safety approval of the FDA, EFSA, and other worldwide authorities. And perhaps the most important topic of all is that you want to be consumer approved. I think that if there is one topic which is the biggest issue, is to be consumer approved. You don't want to sell to the Whole Food and the premium market. You want to sell to the dollar store, and you want everyone not to be afraid of the new food. And we see in our eyes that with such foods, people are accepting it. And while, for example, insect food in some countries has a yuck factor, in other countries it's readily available, the same is going to happen with this, because we do have a need. But in order to accelerate consumer adoption, we must put a lot of emphasis on consumer approval. And last but not least, we want it to be sustainable, again, with a new standard compared to food which is coming from the farm. And waste management and carbon footprints are definitely issues. And after we do all of this R&D and build our companies, we want to have some period of time at least where we can be protected as to the intellectual property, so others don't immediately replicate what we do. And just to have a line of beginning, David already alluded to the excellent report of Rethink, the best report in the market. It's very optimistic, more than I am. And what they are claiming is simply that we are moving from one fermenter to the other. We are moving from the fermenter of the cow with 4% efficiency to the fermenter, which is industrial, with 40 to 80% efficiency. And this is the bottom line. Anyone who wants a comprehensive overview of the precision fermentation, the link is found here, you can follow. When we move to stability, we need to fit proteins for the market. And nature is doing exactly the same. For example, photosynthesis on one hand is going on with this protein, the photosynthetic reaction center in Antarctica, and on the other hand, it's going on in hot boiling springs such as in Yellowstone in cyanobacteria. Well, my PhD was focused on this, and I succeeded to take a protein which is living in cold environment, change a few amino acids and fit it to a warm environment. After publishing what is now the main book in the field of computational protein design, I thought that this method is fit to cure our food system, and that's what we are doing now in Amai. So the bottom line of this is that within a fermenter, even if the microorganisms can grow in high temperatures, which has some benefits in storage and all of that, you have to produce a functional, non-denatured protein. Otherwise, you cannot handle it, even if later you're chopping it into pieces. And you have to think about the temperature it undergoes in the fermenter, in transportation, and in food processing. Now, just to take as an example, sweet proteins are found in nature and are readily known, no glycemic index, no calories because they're hundreds to thousands of times sweeter than sugar, and they are healthy, no effect on microbiome, liver, and kidneys, unlike other sweeteners. And if that's so good, how come we are not using them? That's because of issues of cost, stability, to temperature, to acid and to fat environment, and a lingering taste. What we are doing at AMI, and this is coming from an external report by one of our large company collaborators, we made the protein 90% cheaper than sugar by making the world's sweetest protein, stable to pasteurization with long shelf life, with a sugary-like taste, and with good synergy even with other sweeteners such as stevia. Just a couple of weeks ago, I was supposed to be in Future Food Tech San Francisco to open the meeting with a sponsor of the first session of Ocean Spray to announce our protein-sweetened cranberry juice, which is very good, and people cannot distinguish it from the sugary, full sugar product. And the same we have with ketchup, 70% reduction in other products. So to move from stability, which is a big issue I only touched a bit, into high yield expression, the preference is to express in microbial systems. I will not touch here mammalian systems, which is another whole new ball game, which I don't have the time to enter. And just to give a few examples, this ocean pout fish lives along the shores of Canada and New England in very cold water. And this soybean lives in places where there is an issue of nitrogen. It has to fix nitrogen through its roots, and this cow needs milk. You have here a few proteins which we need. The ice structuring protein, which enables to be in a very cold environment and catches the nuclei of ice without causing freezing, without causing small drops of ice. You have the soybean, which uses a leg hemoglobin, which David already alluded to, and you have in the milk of cow, you have whey proteins such as a beta-lactoglobulin. The ice structuring protein is already produced for many years in baker's yeast by Unilever and is placed in ice cream. So during transportation, when you have partial melting, you will not have an issue of getting small seeds of ice, and you will keep the smooth texture. The leg hemoglobin is produced by another yeast, the Pichia pastoris yeast, by Impossible Foods, and that's their secret sauce. And the beta-lactoglobulin is produced by Perfect Day in a filamentous fungi, Trichoderma raceyi. All of this information is public domain if you go to the FDA GRAS notification database, and you have here the numbers and the years for each of these approvals, and you can learn a lot about the process and what was done, because the information for the FDA is largely, not all of it, but largely public domain, and the same you can do with EFSA journal, which publishes the approvals of novel food to these proteins in their side. So these are just a few examples, but to the best of my knowledge, while chymosin of rennet is the most known protein that is used today practically in almost 100% of cheese production and is done by fermentation, these are the only proteins which are ingredients and not processing agents, meaning they are part of the final product and they're not just used and thrown, and consequently they are much more evident to the final consumer. So that's a category which is very different from processing agent. And then you need to make them in large amounts, and you want to optimize their production in a food-grade host organism. And for that, you need a microorganism that is what's called strain developed. You want it to be a factory only for making your protein. A yeast, for example, must grow in many different environments, for example, an environment with heavy metals. So it has a whole mechanism to throw away heavy metals, But if you grow it in the fermenter, you spoil it and you hug it and you embrace it and you tell it, don't do anything but my protein. Don't do all the rest. And for that, you don't need the entire mechanism of heavy metals and excretion from the yeast. And this is, of course, only one example. Amai is working with many academic labs, which is very cost efficient, and I think is a win-win situation also with the academia. We are working practically with many of Israel's academic service units, including Gil Jonah, and thank you for doing fermentation for some of our needs, fermentation that yielded proteins that are now being tasted around the world by large companies, and that was just the beginning. Since then we did much, much more. And you have a bunch of companies, of which only a handful is listed here, that do such processes. However, some of these companies are yet not fit to work with startups. Some of these companies told us, well, you give us a sequence, we will work on it, but then only we produce it. We cannot work that way. Some companies actually wanted to buy us and to move us, or buy a specific protein and to move it into their system or get 50% of the company or the profits of that protein because they see themselves as partner. This is part of a revolution. Different companies have here different systems. There's also a different issue as to food grade and optimization methods, and I'm happy to continue and discuss this offline because I think this is an important issue of nurturing, possibly with funding from GFI or others, better collaborations between the different entities which can help in the production of proteins by fermentation. The next issue is antibiotics and contamination. And here you have something a little bit more technical. The common practice in the field of what David calls recombinant proteins, what I call proteins produced by heterologous expression, is to use antibiotic resistance. When you clone the gene into the new microorganism, you also clone a gene for antibiotic resistance. You let the microorganism grow with that antibiotic, and only those who can grow, accepted your new gene. But then you have an antibiotic resistance gene in your cells. And also, if you want to maintain it and be sure there are no contaminations from other things, you continue and feed it with antibiotics. To solve this, one option is you need to be much more sterile, but to use an auxotrophic strain, such as a strain that cannot produce an essential compound like the amino acid histidine. And then instead of putting antibiotic resistance, you put the missing gene that will enable it to produce histidine, and then you grow it in a medium with no histidine, and then you know what grew and what not, and you solve this problem. However, this has a lot of disadvantages, and an alternative, which we are using in some of our work, is after you have the initial screening with antibiotic resistance, you can use CRISPR in order to remove the antibiotic resistance gene. In addition, and going into contaminations and antibiotics, we know that meat on the shelf will get rotten, and if the meat is not in a tissue, even much more, because you have much more of a surface area to volume ratio. So you need to be here very careful with sterility, and this is a big challenge of the field. Also, during purification, you must be sure that you do not put toxins from your host organism, and you need to know which host organism you use and what systems you use. In our case, we spent quite a lot of money doing some fermentation in a place where we got a lot of pellet, and it's now sitting in the freezer. We are not eating it because we found out in that place they're also working with other host organisms, and we were afraid that some toxins will be there. Of course, you can check it, but this is another issue in the practical side you need to be aware of. Another topic here is that during purification, what is common practice in the academia, such as using nickel columns for affinity chromatography and so on, you need to make sure that from the purification process, you don't have any, in this case, heavy metals or other things that will contaminate your final sample. The final sample must be food grade. When we did some fee-bearing agreements, first agreements with some of the world's largest companies, they paid us, and most of the money actually went in order to show in FDA accredited ISO and so on labs, external labs, that our samples are pure, are clean, have no contaminations. The money at this R&D stage to pay for that was much more for the actual production by fermentation. Then we have the issue of being affordable and being cheap. And for that, choosing the right host organism is very important. As you go to a less sophisticated organism, not mammalian, not eukaryote, prokaryote, you get a cheaper and cheaper production. If you can go with anaerobic, you save the oxygen that goes in, and that's another way to make things cheap, but you need to be food grade. I will not go into the media, which David touched upon. I'll just say, when you purify the protein, and this is another issue where we had discussions with some big companies, you should purify it only to the level that you want it. But it is not pharmaceutical. You do not inject it into our blood system where you have a severe issue of immunity and other issues. For example, the Impossible Food final product, Rubia, has 28% of non-leg hemoglobin products. At least that's the latest I heard from them. And that's totally fine because those are all proteins we can actually eat. In our case, we do a much cleaner preparation, but we had a lot of discussions on how clean should we make what we do, and we may end up doing different levels of purification to different applications. Whether we want it in a clear beverage or in a granola bar may differentiate what level of purification we do and the cost of the final product. And of course, you need to use scalable purification systems, which is very different than what is commonly used in academia. And in many ways here, you go backwards. You go to tangential flow and to other ultrafiltration and methods, which today you have very sophisticated filters for size, electrostatics, hydrophobicity. In the picture here, we have a small filter that we are using in the labs. The point about this filter, which is in this case a very simple filter by size, is that you can directly and in a linear way scale it up to hundreds of square meters without a problem. This is not the case for columns of size exclusion or affinity or other cases which are not scalable. Now we come to the regulatory issue, and the regulatory issue is a big issue I can talk about for a long time. I'll just say that we all want our stuff to be safe and also to have a labeling of safety. And when we look on proteins, we don't want it to be a toxin, and the anthrax and snake toxins are proteins. We don't want it to be an allergen, and we don't want other toxicity related to digestibility or to hampering with peptide-based hormones, which in some cases you can get, and we don't want toxicity coming from the host organism or from impurities. An important point here is that the FDA and EFSA are taking a lot of action in order to fit themselves to the new foods and ingredients. And I've written on LinkedIn a few articles on this. In June, I was asked by the FDA to join a ISGP, Institute for Science and Global Policy meeting with 86 stakeholders. GFI were there, a lot of VP of Regulatory Affairs from large companies, consumer advocate groups. There were 2 startups there, each giving a 90-minute discussion, and Plato Caucasus and so on. The 2 were Perfect Day Foods, Perumal Gandhi and Ryan Pandya, and myself. Definitely, we are also seeing a changing regulatory framework in this field in order to adapt for Food 2.0. I'm thankful to the GFI for helping me in one specific topic, which is the topic of animal testing. I moderated a session in SynBioBeta about synthetic biology food, and Carrie McMahon was the FDA Consumer Safety Officer who handled Impossible Foods and others, and this is also coming from talks with some of the folks in the different companies, came up and said the FDA never asked Impossible to do animal testing. Once they did animal testing, 90-day rat toxicology, in many places they cannot claim that they are vegan. In our case, one of the world's biggest beverage companies asked us not to do animal testing, and they are actually helping us in doing the regulatory process without this, because simply on their beverage that we all drink, there is a statement of no animal testing. So here's a GFI is definitely taking action, and thank you for doing it. And if you want to see the booklets that came out of this important meeting, the link is found here. Another very important topic towards the end is the consumer approval, because at the end of the day, it doesn't matter what FDA will say, and it doesn't matter what we say, it matters what the end consumer says. And the craziness about GMO, irrespective of my own thoughts about GMO, is a fact. And today there is a question, what is GMO? In our case, we make a protein with heterologous expression. However, in the final product, there are no remains of DNA or remains of the host organism, and consequently, according to both EFSA and the USDA, and the USDA is the one behind it following the FDA, we are considered GMO-free. However, the GMO today, we will not get the butterfly label from the non-GMO organization in the US, and I call here to GFI or to others to issue new labeling about GMO. I think we cannot escape the story about GMO, and we need someone more credible than the private non-GMO organization, Butterfly Organization in the US. Then we have an issue about naturality. Is a novel protein natural? If we changed a few amino acids, we still have a protein produced by a natural host, which is a natural microorganism, but it is a new sequence of amino acids. Is it natural or not? I'll just keep the question. The same goes with the perfect day beta-lactoglobulin, which has a different pattern of glycosylation. It has the same sequence, but the glycosylation is in different places because it is done in a different host organism. Is it natural or not? Is it nature identical? Actually, they use their pattern as part of their IP. And then there is the issue of novelty. I would like to finish my talk with the issue of language. And Dr. David Welch, let me be here politically incorrect and provocative and tell you that I don't like some of your dirty language. You used the word recombinant protein. I don't like it. I think that the word recombinant protein is something frightening for the population, for the consumers, and I would like to move to heterologous expression. I would like to see words such as mutation move to substitution, and engineering move to design. And this is not just something to joke about. This is something that at the end of the day will say how much will be the speed of accepting what we are doing. And the same goes with how to call fermentation-derived food. Is it lab-made? Is it cultured? Is it flora-based? Perfect Day Foods called their food flora-based. They consulted with many people, myself included, and they thought that flora-based will be the best wording. But wording here is important. We do not want the consumers to think we are making Frankenstein things, and we do not want them to be frightened from the food we make. And at the end of the day, we're actually doing something which is much more healthy than what we had so far. And just from my paper in this booklet, I think we have a disruptive beneficial innovation, but we still have a lot of challenges that stem from the consumer acceptance and have a lot of things involved in it. And I think we have the tools to face it. It's just like hospitals. In the past were a hospital for the sick, and today our health centers, the same way we need to move here. I will not get into the details of the solution because I'm out of time, and I'll just end by saying, without getting into the details here due to lack of time, that waste management is also an issue today. For example, when you make a beer in many countries, because a yeast is a GMO yeast, they actually burn the yeast and not even give it to feedstock. And this is another issue we need collectively to think about how we change. With this, I would like to end and to say that we have success in curing some diseases. With the corona, we see that it's not a full success, but we need much more success in curing the food. And the way to do it is by fermentation. Precision fermentation is here to stay and will be used to change the food systems that we have today. Thank you.
Thank you, Ilan, so much. This was really fascinating, and I think the questions you raised are really important. We have almost 130 participants and only several questions. So if you have any questions for David and Ilan, this is the time. Ilan and David, I will allow you some time now to go over the questions, and I will share with our audience a few slides about what GFI is doing here in Israel. So first of all, we are a relatively small team. Alongside myself, we have Or Benjamin as the Director of Operations, Dr. Tom Ben-Aryeh, our senior scientist that many of you know, Hadas Karshai, our macro manager who brought all of these events to life, and our executive assistant, Lior Schneidman. We are hiring nowadays for 3 more positions. So if you or someone you know are excited about what we talked about today and alternative proteins in general, please take a look at our openings at the Academic Relations Manager, Business Engagement Manager, and Policy Manager. You can feel free to reach out to us either through email at israel@gfi.org. We are also available on Facebook, LinkedIn, and soon on YouTube with all of the seminars that we just had this couple of weeks at the Good Food Institute Israel. The Science of Alternative Protein series is almost coming to an end, but we have an event on April 6th with Tel Hai College about the implications of the coronavirus on the Israeli food tech ecosystem, and I will share the link to it with you in a minute. And now let's go back to the Q&A section. Let's see. So we will take it from the top. Ilan, you want to say something?
I can start answering a few questions, or do you want to read the questions and then us answer? What do you want?
Let's, I'll try and moderate and begin with the first question from Alon, who is asking David. David, can you please elaborate a bit on the pros and cons of anaerobic bacterial fermentation versus aerobic fermentation for the production of biomass?
Yeah, I can start, and I'd love for Ilan and maybe Gil as well to jump in with their thoughts too. So aerobic will be lower cost most likely, and the positive is that there are already facilities to do this at large scale that are used right now in the like for bioethanol production. But there's some challenges around sterility and suitability for food grade ingredients, and Ilan touched on some of this in his presentation as well. But I think this also presents an opportunity to identify or optimize host strains that can grow in conditions that are unfavorable for contaminants. So companies like Air Protein and Calista and Nature's Find, which used to be sustainable bioproducts, are examples of this that have identified microorganisms that grow under very specific conditions that could solve some of the challenges in aerobic fermentation. Ilan, did you want to add to that? You're on mute. There you go.
A good friend, and we are actually co-located in the same space. I think I will pass this question. I think there are a lot of advantages to anaerobic fermentation in some cases in large scale also.
Okay, thank you both. Moving on to
One,
Thomas.
I have one more skill. So another advantage using anaerobic is the fact that you don't get oxygenation of protein, and therefore it may help prevent all kind of damages to proteins.
Very good point.
Perfect. Thank you. Thank you all. Thomas is asking, does casein need to be folded in order to produce cheese? Often the folding, he says, of a protein is the most difficult part in producing proteins. David, would you like to take it?
Yeah, I'll say I'm not a dairy protein expert, but my understanding is that at least in the micelle structure, the casein proteins are unfolded, but there's folding events that can occur depending on the conditions during milk and cheese production. So folding is not a requirement for micelles, but folding is an event that can happen. So we need to be conscious of that when we're producing proteins through other methods.
Just to add on top of that, I fully agree with you, David. Casein is a very important protein. However, it is unique in the fact that it is generally unfolded. 10% of protein spaced out intrinsically unfolded, and casein is one of the known cases of a whole protein for that. This also limits our ability to produce casein in other host organisms, because when you produce it before it goes outside the micelle and so on, if it will be unfolded within the cell, it will cause aggregates there, and then we should have to produce. So what we do actually using computational protein design is actually look at areas which are partially unfolded and try to refold them, or in the case of loops, to loop rebuilding. But in the case of casein, it's a very different ballgame.
Thank you, Ilan. I'm not sure if everybody could hear you well. I had a bit of a hard time. Let's move on to the next question. What about GMO status if fermentation is with GMO fungi? David?
I'm going to leave that one to Ilan. He's the GMO expert.
So I'll combine this with another question here. And we had many talks about the GMO status with FDA, with EFSA, and with consultants that we have of these, and also with some of the other companies. And I've spoken with some of the companies I presented here with our VP of Regulatory Affairs and so on. I want to make something very clear. Just like Perfect Day and Impossible Foods are not hiding the fact that they are making a protein by heterologous expression, so do we. However, once you have a plant that is transgenic, then you actually eat the DNA. And if you will ask most of the people in the world, are they willing to eat DNA? Most of them will say no. People don't have a clue, the general population, about those terms such as DNA and others. From the point of view of the European Food Safety Authority, and I'm willing to share this legal information with anyone interested, you have a track for GMO and a track for non-GMO stuff. Unlike the FDA, they actually are looking into the GMO character of what you make, and it is stated there very clearly if you do not have remains of the host organism or remains of DNA in your final product, then the GMO which you did use, and in our case it's not GMO but GMM, genetically modified microorganism, the GMM is considered legally a processing agent and is not part of the final product. Actually, you cannot call the final product a GMO because you don't have there a DNA footprint. A recent work that is by the USDA that was with a lot of lobbying and talks by the Industrial Biotechnology Organization, and Vincent Seewald from DuPont was a key character there, heading it for a long time, is today the same applies also for the United States. You do not need to register it and do anything related to GMO as long as in the final product you don't have remains of the host organism. However, from the labeling point of view, in different countries and different places, you will get different definitions of whether you can call it no GMO or GMO-free. And I alluded to the fact that in the US, extreme organization, private organization that is controlling what all of us will eat, and I hope GFI or the FDA will change that and issue something more credible about the GMO status of things. Irrespective of my own thoughts, that GMO today is not something that is bad for it.
Thank you, Ilan. Before the next question, I just want to point out that you can also ask questions to Gil or Ronnie, maybe about the Israeli Fermentation Society. The next question is from Shulamit. She's asking, she's saying that we talked mainly about taste and texture, but nutritional qualities were only mentioned concerning animal feedstock. How can the nutritional values of meat and milk be expressed in the fermented engineered food?
I will take that. This is a very important question. We actually have today a pipeline, a bunch of proteins which are basically weighing funding, and we'll move to work on them. We did some work then. Some of them we have a specific issue of making proteins which are more nutritional, with more essential amino acids, something which is especially important, for example, in baby food, in infant formula. And the idea is that if you don't need to add essential amino acids, you can make food which is more nutritional. In this sense, proteins in general are nutritional. Amino acids are good for you unless you really consume huge amounts, which is something sometimes done by people who are into sports and eat too much protein, whey protein mainly, powders, and then they can have a accumulation of uric acid, which is takes time, and then it gives you pain in the joints. But in the regular amounts, all proteins are good for you. You can add and make a protein or a whole protein or digestible protein by changing the amino acid profile of it. And in this sense, science of computational protein design is focused most often not on the binding site or the active site, but rather as the scaffold which holds it, which is generally robust to changes, and there it is more easy to do such changes in order to make the protein not only more stable, but also more nutritional. Definitely an important topic for both food and feed.
Thank you, Ilan. Unfortunately, we're having a very hard time hearing you. Maybe you should try not touching the microphone itself. David, would you like to comment on this question?
I think Ilan covered it really well. I think the point of getting the amino acid profile right, the digestibility score, is going to be more and more important in these alternative protein products. Initially, the market is focused a lot on taste and texture, but I think the question raises a good point that the industry as a whole doesn't just need to focus on getting the sensory profile right. It needs to optimize the nutritional profile as well. And I think that's really the power of this industry, right? The flexibility that all of these ingredients, whether they be fermentation derived or plant derived, provide. We can assemble them in the right way to create not only the perfect organoleptic experience, but also the ideal nutritional profile as well.
Perfect, thank you. Let's direct another question for you, David. Again from Shulamit. Can you please explain how the new food is more healthy than what we haven't had so far?
Yeah, this is a really good question. I think that, well, I refer you back to the answer that Ilan and I just gave in terms of, you know, how we can optimize the nutritional profile through, say, amino acid composition. But also, I would think about this from a global health perspective as well, right? So I spoke about in the beginning of my talk about how rampant the use of antibiotics is in the industrial animal agriculture industry. And by shifting away from a reliance on that type of industry to a more sustainable food system that uses fermentation ingredients and plants, we can remove the need for that overuse of antibiotics that is causing these really critical issues with antimicrobial resistance and superbugs, which is really going to impact the health industry in massive ways, and society as well in the future. And then, you know, we can think about foodborne diseases and pandemics, and how many of those are connected to the way that we use animals and the way we contain them in close captivity for food applications, and how this industry we're speaking about can resolve some of those really big global health issues, while at the same time optimizing these food products we're making from plants and fermentation ingredients to get a really perfect nutritional profile.
Thank you, David. Let's see. Carmen is asking, how promising would you consider algae as meat alternative? An example when it comes to making a bacon flavor.
I think it's yeah, it's really promising. I think there's some questions to solve in terms of how scalable algae is, and how quickly we can bring down the cost relative to other microorganisms. But I'm really excited about what can be done with algae and seaweed. We're actually funding 2 projects through our competitive research grant. One of them is actually in Israel, to look at novel ways of using seaweed ingredients for this industry. So I'm very optimistic about algae, but there's some cost and scale questions that exist today.
I would just like to add, I'm also very optimistic about both algae and microalgae. There are different issues. One is to use a final algae as is, with very minimal purification in the final product. The other is to use it as a mechanism to express proteins, whether they are...
Ilan, I'm sorry, we cannot hear you well. Maybe you should try turning off your video. Maybe it will help with the quality of the sound. Let's try again.
Okay, I'm sorry for that. Do you hear me now better?
Yes.
Okay, so algae is definitely a very important host organism. You can use it either as a final product, or you can use it to express proteins. Then you have the issue where you can clean the algae fully from the protein you export.
Ilan, I'm so sorry, but we cannot understand the word. I apologize. Maybe Ilan, you can take a look at the questions that you have in the Q&A, and you can actually type the answer if there is a question directed to you. And whoever submitted the question, you will be able to see Ilan's answer
Okay,
in the Q&A section.
I'm moving to a different Wi-Fi, which will hopefully be better.
Now, okay. So let me know when you are ready. Let's see if we have any questions for David. David, did you see any question that you like in particular?
I saw a question about scaffolds, which I'm now, oh, there we go, from Gary. He asks all of us, I'm interested in scaffolding cell growth. Is scaffolds a big issue with cultured meat regarding what's available and the costs? Yeah, this is a really interesting and good question. I think there's a lot of work right now to try and identify scaffolds that work well to culture animal cells in a three-dimensional environment, and do so at low cost with ingredients that are food appropriate for food. So there are, you know, a number of polymers and other materials that are used in the cell therapy industry to create three-dimensional tissues. But many of these are either not approved for food applications or would be difficult to get approved. So there's a lot of interesting work beginning to look at using algae and plant materials to create scaffolds. I think you know, fermentation can come into play here when you think back to the growth factor question as well. And potentially, rather than using growth factors in the cell culture media, we could actually attach them to these food relevant scaffolds to provide the instructions that cells need to grow and divide and differentiate. So lots of white space opportunities for scaffolding materials for the cultured meat industry.
Perfect, thank you. Ilan, how is the new Wi-Fi?
I hope it's better.
Let's see. Is there any question in particular you?
Yeah, there is a question by Lucilla about allergenicity. And this is a complex question. From the point of view of the FDA, we can do a in silico bioinformatic analysis, which is sufficient. If you look in the case of other proteins, which I alluded to in the past, in some countries, they have undergone additional allergenicity and digestibility testing. And we are working with a bunch of different organizations and companies to do that. But the initial allergenicity test, it's enough to do something which is 100% computational. And I'm happy to send anyone who is interested the protocol, which is already found on the GRN database for some proteins, and anyone can follow to check the initial allergenicity. I want in this case also to mention that there are a bunch of organizations, research organizations, which are very applicative, whether it is A-STAR in Singapore, CSIRO in Australia, or VTT in Finland, or Fraunhofer in Germany. And in all of these places, you have some very applicative work, including on these topics, and we are working with some of them.
Excellent, thank you. And we can hear you great. Now let's take a look at the question from, sorry.
Yeah, Nir, yeah, I see a really nice question from Alexander, which I think Gil could probably answer really well for us, if you wouldn't mind, Gil. He asks, assuming the goal of these discussed fermentation technologies is to one day be performed on industrial scales, what do you see as the most economically feasible primary sources to use as feedstocks? Sugar-based, lignocellulose, industrial co-products, etc. Gil, I imagine you have some thoughts in this area.
Yeah, so in the US, most plants are really near the fields. So any cheap crop and remaining of crops, whether it's a lignocellulosic or byproduct or glycerol or glucose, should be the way to go. So any remainings that you have, that you can use, should be the way to go.
Great.
Nir, with your permission, I would like to ask a bunch of questions which were specifically for me about sweet proteins, but in a much more general way. Brian asked, why did they choose sweet proteins? And Zvika asked about the alternative of saccharin and other high-intensity sweeteners. So we had a very big dilemma, and using this technology and actually this book on computational protein design, which I put out, what to work on. Should we work on disease? Should we work on meat alternatives? Should we work on sweet proteins? And it's the biggest impact on the world, and it's the biggest place where we have a competitive edge as to the technology versus other technologies is in this field. And here, when you choose what protein to work on, you have to take into mind where you can give something which others still cannot. And the same goes with alternatives. Saccharin and other high-intensity sweeteners were designated by the American Heart Association a couple of years ago, and by many others, as things which hamper your microbiome. And actually, a diet cola will make you fat and diabetic, despite the fact that it is 0 calories. So we have here also a competitive edge, not only as to cost and sustainability, but also as to health. I think that with meat alternatives, you have the same issue. If you go to milk, and this was done by Perfect Day Foods, then only 3.5% of the final product is the actual protein. That's number one. Number two, it doesn't matter if you have a different glycosylation pattern. You can actually use it to your advantage to get IP over it. But you do have a competitive advantage, and you can produce large amounts in a very cheap way, and in a very quick way, and in a very sustainable way. When it comes to making a full steak, this is a longer process because you need the growth factors and you need to get mammalian cells in a media which will be cheap. And there we still have a longer way to go. However, the benefit is very big, and you have a bunch of excellent companies in this space. So at the end of the day, just like with any other startup, you need to have a mission, whether it's fully economic or also with some impact, and you need to see where is your competitive advantage as to your technology and as to what you can do.
Thank you, Ilan. We have a general question from Thomas. Thomas is asking, what are examples of topics combining data science and fermentation? Who would like to take this one?
So I'll try to take it. Using computational protein design, on one hand, we got hundreds of thousands of dollars from two known companies, Google and Amazon, and we are heavy users of high performance computing. On the other hand, at the end of the day, on the biotechnology side, we still don't use intensive data science. Ginkgo Bioworks and Motif Ingredients, which is their spin-off working specifically in food, are doing exactly the opposite. They are saying, let's try and use every possible combination of promoter, of signal peptide, of host organism, of strain. Later, let's sequence everything we get, put into the equation also all of the results that we can measure. Let's measure anything we can measure, and let's put it all into a big AI machine learning thing and see how we can optimize precision fermentation. So right now in the market, you have places where the data science is used mainly on the design. You have places where data science is used on the fermenters themselves, fermentation themselves. In our case, using a more rational approach, we found a new promoter, which is a trade secret and which enables a much, much better expression level. And we found it in a totally rational manner, but in a place where no one else has looked before. So you also need to know where to put your spotlight. But at the end of the day, I think data science is an intrinsic part of everything we do. And also in the second phase of production by precision fermentation, where the main driver will be actually cost, than taking a protein which is already being produced and cutting its cost dramatically. Here you will need to see a lot of data science on redesigning or doing what's called strain development to the host organism, and optimizing the promoters, the signal peptides, the entire gene cassette, the copy number, how to feed it in the best way, and so on and so on. This is all data science.
Thanks so much, Ilan. Any comments from the panel? No. So 2 last questions. Shulamit again is asking, how are the new proteins digested in the human body? Any comments on that, digestion?
I think Ilan is probably well placed to comment on this from his from the work he's doing. I'll just add, maybe say one thing before Ilan speaks, is that if you remember back to our presentations, many of these proteins are not novel, right? They're just, we're producing the same proteins that people already eat, but in a more efficient and sustainable way. So they will be digested in the same way as well. But I do think there's more studies that need to be done on these novel proteins and the long-term health impacts, both positive and potentially negative, from them. Ilan?
Remember, a protein is a protein is a protein. At the end of the day, we eat many tens of thousands of different proteins. I have a whole biochemistry course which is around proteins and how they are digested by the proteases, whether it is the carboxypeptidases, the serine proteases, or chymosin. And proteins are all digested in a similar manner. You do have to check that you don't have short pieces of proteins, which are called peptides, which will interfere with endocrinology. And you have to check that your protein is not overstable. And for that, today, you have methods which do not require animal testing. You have ex vivo methods, and you also have methods which are totally in vitro. You can take the main proteases and do an artificial stomach, put a protein into your protein into it, and then see and mass spec everything that is going out. And these are, of course, experiments which we are actually doing together with companies that are experts in this field. Also, you have to ask yourself, what is the final amount that is going to be used? And especially EFSA will give you an acceptable daily intake for each thing that you will apply. And in our case, at least, because a teaspoon of our sweet protein is equivalent to 50 kilograms of sugar, you actually eat very small amounts. So we really don't see any issues with this topic, but obviously we do need to do a few experiments to make sure that this is well evident and that everyone is sure about it. I think that here GFI and others have a role in this changing revolution, and EFSA, a new protocol on how to test.
Ilan, I apologize. We are having a hard time hearing you again. David, would you like to take the last question?
Yeah, I'd be happy to. Rainer asks, how advanced are non-modified mammal cells to produce a cocktail of proteins like milk? And the 2 examples of startups in the space, I think, are the 2, at least that I'm aware of, that exist today. So Turtle Tree and BioMilq. I think in terms of advanced, I think we're at still pretty early stages. These companies have not been around that long. But the technology they're using, or the science they're using, is very similar to that used by cultivated meat companies, right? So they're taking mammalian cells and growing them and differentiating them instead of with the aim to produce muscle and fat to create meat, but in this case to produce actual milk. So the same we can apply the same scientific knowledge to this of how we grow these cells in an efficient manner, the growth factors that we use to maintain and differentiate these cells, and how we scale this up into large-scale manufacturing. These questions are all very similar, and the same types of challenges exist. So how do we bring the cost down? How do we improve the cell culture media so that it's more cost effective? How do we scale up with the right types of bioreactors and other equipment? So I think it's a really promising field, but very early stage, that can benefit from the advancements that have been made by cultivated meat companies over the past 5 to 6 years.
And with that, our session has come to an end. I would first like to thank Dr. Rina Samish from Amai Protein for his presentation and very interesting insights. Again, we are hiring. Feel free to learn more about us at our social media. Our next event is on April 6th. You have the link to it at the chat about the Israeli food tech ecosystem dealing with coronavirus. Big thank yous to our partners here, Gil and Ronnie from the Weizmann Institute of Science and the Israeli Fermentation Society. It was a pleasure working with you on this event, and I sure hope it's the first of many.
Thank you.
Thanks, Gil. And toda, ובתיאבון, stay safe.